Thanks very much for the invite. It's really a pleasure being here. And I think I have to say, it's the first lecture I've been invited to where I get to invent a drink. So I hope everybody's enjoying those mutating martinis. So really, what my lab is focused on is depicted in this slide. And we're focused on various human diseases. And how we try to understand these diseases is we generate mouse models for these diseases that we're interested in. And really, we love our mice, I'll tell you, but it's not that we're trying to understand mouse behavior or physiology. We really would like to use these models to pinpoint the gene or genes responsible for the disease under study. And we'd really like to know more about the mechanism, really the nuts and bolts of how those genes are encoding products that ultimately are responsible for the human disease. So the real goal is to apply what we learned from these mouse models back to the disease under study. And I think as a scientist, I have to say that it's really, really amazing to have one discovery in your career, really. And I've been sort of really lucky and have three basic things that we study. And we've had three major discoveries. And so what I'd like to do tonight is to give you sort of vignettes of some of these projects. We work on P63, and I'll tell you how we came about working on that gene, and also CHD5, and I'll tell you about these. These are both cancer-related projects. But really what I'd like to do is to tell you about some of our most recent discoveries understanding the genetic basis of autism. And as all of us know, there's about 10 to 13 cells that make up the human body. And within each of these cells, of course, there's this genetic blueprint shown here in this chromosome spread. And essentially, we can then take these chromosomes and analyze these, is what geneticists do. And essentially, this is an artist's rendition of one of these chromosomes. And basically, this is showing the genetic blueprint in a single cell. And what cytogeneticists, essentially what we do in the lab is we splat these, it's called the chromosome spread. Somebody holds the single cells and splat them onto a microscopic slide. And here you get this nice display of chromosomes. And then the cytogeneticist, of course, names these particular chromosomes and lines them up. And here you see chromosome 1, it's named its largest chromosome, and down to chromosome 23. And here these chromosomes are painted in different colors to show you their identity. Now here, instead of the normal karyotype or group of chromosomes that I showed you previously, what is shown here is the group of chromosomes from a cancer cell. And so what's been really useful to my laboratory in all three of those fronts that I showed you is the ability to model these type of chromosome changes. So what's here in this cancer cell is some specific chromosome abnormalities. So there's chunks of the chromosome missing, or there's extra regions. And what we can do using mouse models, and I'll tell you more about this technology, we can engineer the chromosomes, and we do this in mouse embryonic stem cells, and then we make living, breathing mice that have the same kind of chromosome change as seen in the human disease under study. So we can make, with chromosome engineering, these are precise rearrangements. We can make deletions where we're clipping out a group of genes. We can duplicate, make extra copies. We can flip it around. We can even move one piece of a chromosome to another. And this has been really powerful for us into pinpointing genes responsible for various diseases. Now, why the mouse? You might ask this. This is a really good question. Again, we really want to understand human disease and health. Well, the mouse actually has physiology that's quite similar to that of humans. We don't in any way think mice are people, but they are very, very useful for pinpointing what goes wrong in various diseases. And also, the second reason is they have essentially the same genes found in humans, and they tend to cluster. If a group of genes are nearest neighbors in human, they also tend to be nearest neighbors in mouse. Now, I think one of the most remarkable reasons, to me at least, why we use the mouse as a model system is that we can engineer these particular changes to understand gene function. So gene sequencing is great. We have a lot of sequencing efforts pinpointing the genes, but we really ask, what does that mean if we clip out a piece of a chromosome, for example? So we're able to engineer any particular change that we'd like. And this is showing sort of the schematic of how we do this, we grow the embryonic stem cells shown here. Here's an embryonic stem cell clone, and it's grown on essentially a feeder layer, which is shown here on this plate. And these feeder cells secrete the nutrients needed to keep those embryonic stem cells happy. And then once we make a particular change in the culture dish, whatever we'd like, then we inject these embryonic stem cell clones, shown here, into a very early developing mouse, and that's shown schematically here in white. So the cells shown here in orange are the cells that we've genetically altered any way we'd like. Then these very early embryos, these so-called blastocysts, are surgically implanted into the uterus of a foster mom, essentially. And out come these chimeric mice, which are a mixture of those cells we engineered and also these host blastocysts. So this chimeric mouse here that you can see by this nice coat color, basically we know this experiment is successful because we have a more pigmented type of coat color, which is made from the contribution of these cells we've altered the way we'd like. So this chimera is essentially like a granddad that when crossed to wild-type mice can transmit those particular rearrangements of a chromosome, for example, or gene knockouts to subsequent progeny, and then we can study the effect on the mouse in vivo. So first I wanted to tell you about one thing we study in the lab, and that's P63. And this gene is really something that's near and dear to my heart because it's something I found when I was a postdoctoral fellow at Baylor College of Medicine, and I was this young scientist. And basically what I was able to find, this is a page from my notebook in '97 actually, and what's shown here is I was able to amplify a really small fragment, a PCR product shown here of 139 nucleotides that was a chunk of this gene called P63. And the reason this was important is because P63 was a new gene that I happened to sort of stumble into actually, that is related to P53 that you may have heard about, which is a tumor suppressor gene it's mutated in about 70% of all human cancers. So it's an important, P53 is an important cancer gene, and nobody knew P63 even existed. So I found this little tiny piece of it, and it ended up being really, really exciting at the time because it was something present in both mouse and humans, and I got to be on TV. I was pretty excited. This was my first.
Researchers have identified a gene that is similar to an important gene that suppresses cancer tumors. The newly identified P63 gene affects limb and skin development in mice. Now, it's from the same family of genes as the P53 gene. Baylor scientists years ago were first to demonstrate that the P53 gene plays a critical role in cancer development. So scientists theorize this new gene may also be involved in cancer development.
P63 is a new gene that we've isolated, and it has homology to the P53 tumor suppressor gene. And that's a gene that's been found to be mutated in about 50% of all human cancers. So
now the focus of these Baylor scientists will be on determining if and how the P63 gene may be involved in cancer growth.
So that was actually our question. So this piece of the gene I found looked a lot like P53. What does it do? And I happened to be in this laboratory where we were able to manipulate the mouse genome really very, very efficiently. In fact, the guy who trained me, his work when he was an undergraduate won the Nobel Prize for his boss. I was pretty happy about that. My students were less happy when they found that discovery. They didn't think their thesis work should contribute to my Nobel. But nonetheless, so in Alan Bradley's lab, I found that if you don't, I knocked it out. I made mice that didn't have P63, and the effect was really different than what was known for P53 deficiency. Essentially, what this found is that if you don't have P63, you don't have arms or legs or skin or hair or nails, mammary or prostate. Everything's pretty much gone, and you die as a newborn. And this is showing a mouse phenotype where there's limb defects you can see really clearly. And this was really important to other clinicians that were looking for the cause of these developmental disease syndromes where the children essentially had problems with their hands and feet and craniofacial clefting and skin problems. And sure enough, after we found this in the mice, these clinicians then said, okay, P63 looks like a great candidate. And they looked carefully at P63, and sure enough, there were point mutations in the p63 gene responsible for this human developmental syndrome called EEC. So that was really pretty nice. And what we were able to show more recently in my lab at Cold Spring Harbor is p63 is important in organismal aging. It's actually important for replenishing your stem cells throughout your life. And if you don't have very much, you grow old at an earlier stage. So that was really interesting. And then also, so actually, I should point out, when I first found this, that p63 is essential for development. A lot of the p53 cancer world said, oh yeah, but that's not cancer, that's development. And they didn't think that was very important. But actually, what we've been able to show, development is very critical in how the stem cells replenish. And what we found is some versions of p63 proteins are tumor suppressors that can shut down the growth of a malignant tumor in vivo. So this was pretty important because, of course, if we could do that clinically, that would be really fantastic. So the gene encodes these cancer-preventing genes, these tumor suppressors called the TA-p63 proteins. But also, interestingly, the same gene turns on other proteins that cause cancer, these delta N p63 proteins. And these are cancer-causing because they make the stem cells proliferate out of control and trigger cancer. So one gene is encoding proteins that both prevent and cause cancer, and it's all about cellular context. It depends what cell type these are expressed in, and hence what tumor types form. So the p63 discovery has really grown into a whole field of its own. It's been really exciting. We now even have a workshop just on p63. It's always in France, so it's fantastic. And the developmental studies really now have been expanded from that single human syndrome that was first shown to be caused by p63 mutations to now it's known that p63 mutations are responsible for 7 different human developmental disease syndromes where the kids have, for example, limb problems and craniofacial clefting and skin issues. So this is showing kind of a subtle phenotype. You may say, okay, that's not a big deal. The fusion of the toes or the digits, it's not that bad. But there are other cases that are much, much more severe, shown here where you can see severe clefting. And so it's a really serious issue, and clinicians are even doing in utero testing for families that have had one child born with this and 50-50 chance of having another one. In addition to being the cause of these human developmental syndromes, essentially every pathology lab throughout the world, I'll bet, is using p63 as a marker to classify various tumor subtypes, such as skin cancer, and also just this week there was a certain classification of urothelial cancer affecting the prostate differentially. So p63 is an important marker used to diagnose cancer. So that's one thing we've worked on, and CHD5 is another quite exciting project. This is more recent than the p63 work. And what was found, it really goes back a number of years, over 30 years, when this guy, Garrett Brodeur, described a deletion on the short arm, the P arm, of human chromosome 1. And this was in neuroblastoma. And this, of course, suggested that a tumor suppressor mapped to this region, and in particular, this region called 1p36, which is sort of the map location of the short arm of human chromosome 1, was commonly deleted in lots of different types of human cancer. So this was sort of this, I'd say, holy grail of tumor suppressors, people trying to find this tumor suppressor because it's not just affecting one type of cancer. It's this chromosomal region tended to be lost in many different tumor types. And this is showing a review a few years ago that we prepared and showing this region called 1p36. Here's the short arm of that human chromosome 1. The long arm would be way over here in this room here. And basically what these deletions here show is the different color lines are showing deletions in epithelial cancers in green, these neural and brain-associated cancers shown in blue, and even hematopoietic malignancies like lymphoma and leukemia tend to have deletions in this 1p36 region. So certainly something was going on in this region. However, it was really, really hard to pinpoint, if you line up all these different deletions in these different cancers, it wasn't as if there was a single region that would pinpoint the gene responsible. Because what I didn't mention is there's hundreds and hundreds of genes along the short arm of human chromosome 1 in the 1p36 region. And so we really needed a way to say, okay, where is the tumor suppressor? It was not at all clear in the literature. And here is where we asked, I think, really straightforward questions. Which region of 1p36 was functionally important in harboring a tumor suppressor? Secondly, once we pinpointed a region, which gene or genes was responsible in coding a tumor suppressor that was preventing cancer? And of course, how does it work? So here is showing the various cancers I showed earlier and all these various deletions. And these cluster in 1p36. And importantly, for this technology to work, all of the genes that cluster at 1p36 in humans also tend to cluster together in the mouse genome. Happens to be mouse chromosome 4. So what we were doing is using our embryonic stem cell technology and our chromosome engineering, we were making series of chromosome rearrangements shown here schematically. And then we do this in embryonic stem cells, and then we make mice that are actually deleted or duplicated for these various regions to see, can we pinpoint a tumor suppressive region? And this was a fantastic way to go because, of course, mice are like us and they inherit one copy from the mother, one copy from the father, and so they had 2 copies of this particular region corresponding to 1p36. And when we made mice that had too few copies, so these have a deletion of one of these regions, so they have one of these copies intact, it was great because cells from these mice proliferated out of control all over the dish, and mice that had only one copy that had this deletion developed a number of spontaneous cancers. So that was really great. But for me, when I was really convinced was when I looked at the mice that had an extra copy. Because we can, with chromosome engineering, not just delete and have a loss of gene function, we can duplicate and have gain of function, so we can directly compare and contrast, losing this whole cluster of genes and gaining it. And here, instead of getting cancer from having too few copies, these mice died right around birth because they had too much tumor suppression. Essentially, the cell division just shut down, and they had too much apoptosis of programmed cell death, and the embryos didn't survive after birth. And so this allowed us to pinpoint this region corresponding to 1p36 as having a potent tumor suppressive activity. So that was great. What I didn't tell you actually is there are 52 different genes in that interval. So we had a lot of genes to go through. Which one is the tumor suppressor? And actually, we didn't even know if it was 1 or 50 of these genes. It ended up being a single gene within the interval that was responsible for this tumor suppressive effect, and that was called CHD5. It happens to be a protein, it encodes a protein that is responsible for how the chromosome, the DNA, is packed. In a single chromosome, actually, if you line up the DNA, it's 3 meters, but about 10 feet-ish in length, and it has to be really tightly compact within that chromosome structure that I showed you. And CHD5 is predicted to do just that. And so we're looking at that in more detail to get in more mechanistic insight. Really, how does it work, and can, of course, we fix it? What we do know so far from our cancer studies is CHD5 is a protein that we think about as a circuit breaker, sort of a switch that turns on by regulating this gene locus here called the INK4ARF locus. This is basically allowing expression of this tumor suppressive network so that when we need it, CHD5 is turning on this tumor suppressive power within our cells. And these are other tumor suppressors that are well characterized, for example, P53 and these others here. And CHD5 is this master switch, and again, I always think of this as a circuit breaker because you need this circuit breaker to turn on all this tumor suppressive power in these cells. And clearly, what we found is if you obliterate CHD5, you cripple this tumor suppressive locus, and then cancer develops, and many different types of cancer, and we're studying that more in detail. Now, this locus has previously been linked to, it's a very important tumor suppressive locus, and so turning this tumor suppressive power on is good because it inhibits cancer, and also this keeps stem cells from renewing when they shouldn't. So it keeps cancer in check. And CHD5 is a regulator of this whole network of tumor suppressors. So this is showing the region of mouse chromosome 4 where CHD5 resides, and there is this 4.3 megabase interval, and 52 genes are encoded here. And all of the genes that cluster within this red region cluster together within the human 1p36 region, within this green 5.7 megabase interval. So all the genes that are here in the mouse are also nearest neighbors in human. But again, we really want to not just pinpoint tumor suppressors in the mouse. We want to see what is happening in human cancer. So I called up my friend at Stanford University, and he was studying human glioma and looking for deleted regions in human glioma. And he is the head of neuropathology at Stanford, so he's a guy actually you don't want to meet. And whenever I go and work with him there, he'll be taken away to analyze somebody's tumor, literally, and he's brilliant. And so he had been studying deletions of human chromosome 1 in these patients with glioma, and really gratifyingly, he came up with this completely independent of our mouse work. He uncovered this 5.4 megabase interval that overlapped beautifully with this human correlate of the mouse region we had pinpointed. And most importantly, of course, is that this overlapping region that we had identified and he had confirmed in human patients is encompassing the CHD5 gene. And then just taking you back to those deletions that I showed you from the literature from many different laboratories in that review, affecting epithelial cancers, neural and neural-associated cancers, and lymphoid cancers, CHD5, the gene we pinpointed, does map within a lot of those deletions. Of course, there's some that do not overlap, so there could be other things going on in this other region of the 1p36 region. So it's really, really fun, I have to say. We have a great time when we find something where Basic Cancer Research Center at Cold Spring Harbor, it's really a phenomenal place if you haven't been there. You should check it out. It's really a great place to do science. And what we have is, we make basic cancer discoveries. We're NCI designated Basic Cancer Center. We don't see patients. But what we found in the mouse is that CHD5 is a potent tumor suppressor, and then lots of people across the globe have been now finding mutations that take out CHD5 in their favorite cancers. And I show this, we update this slide almost weekly because there's many different human cancers shown here that have these particular point mutations, single amino acid changes in most instances that are smattered across the CHD5 locus. So it's really, really fun every time I open up a journal and see, okay, there's another cancer that is upsetting CHD5. Now, a lot of you have probably heard about these large-scale sequencing efforts where people are sequencing various cancers in this case and sequencing the whole genome, and it's great. And we talk about whether these different mutations are driver mutations that are actually causing cancer, or are they coming along for the ride, or are they passenger mutations? And the truth is we don't know yet. These are found most of the time in a single cancer, in a single patient. We don't yet know if they're functionally important, and we have ways of testing this now. But what we are seeing is some of the types of human cancers, for example, breast cancers, are developing in our mouse models with compromised CHD5. So I think it's an important question that we'll look more into. And perhaps most gratifying is some recent work, again, by other laboratories with clinicians who have found that CHD5 levels predict the success of anti-cancer therapy. So basically, these are patients that came in with tumors. This is showing neuroblastoma, And it's also more recently been shown in patients with ovarian cancer, they would have gotten whatever treatment anyway, and the anti-cancer therapy. And what the clinicians have done here is they've categorized the patients according to low CHD5 expression in the brain for this neuroblastoma case, and then those with high levels. And what they find, importantly, is basically you want to be here because if you look at the percent survival after the treatment as a number of months, I know it's kind of hard to see here, the patients with more robust levels, shown here in brown, of CHD5 expression live much longer than those with lower expression, shown here. So I think it's really exciting that CHD5 is being shown to be a prognostic marker for survival after anti-cancer therapy. So I really want to tell you about some of our recent work, again using chromosome engineering, and really what we wanted to do. We had this success with cancer gene discovery, and then we thought, why not apply this to other human diseases where it's known to be a copy number change or a chromosome abnormality? And so we started working on autism, and this is a very recent development in the laboratory. And so basically what I'll show you kind of briefly is we use chromosome engineering. We've made this mouse model of autism, so I'll tell you about how we've generated these mouse models and how we've analyzed the behavior of these mice, and also how we've pinpointed alterations in the brain architecture in these mice, which we think is very important for understanding autism. And so autism spectrum disorders, or ASD, are characterized, they've really been popularized by this book here, The Horse Boy, and also there's a movie. And these patients are characterized by three cardinal features: altered social interactions, there can be language impairments, and repetitive or restricted type of behaviors. And there also can be things like sleeping difficulties and sometimes eating problems in these children. And it's one thing, talking to clinicians, is one thing that I think clinicians do agree on is that ASD patients vary widely with regards to their abilities, their intelligence, and behaviors. And sometimes even children in the same family have very different clinical manifestations in this diagnosis. And so how did I get into this? Well, my colleague at Cold Spring Harbor, Mike Wigler, had published this paper in 2007, and he found this strong association of these chromosome changes, these copy number mutations in children with autism. So again, pieces of the chromosome missing, extra pieces of the chromosome where they shouldn't be, things like that. And we started talking, and actually one region that he found was this 16p11.2 region, again, on the short arm of human chromosome 16 in this case. And what he found was there was actually a single patient at the time, happened to be a little girl, who had a deletion, missing a cluster of genes in this region. And so we had the tools, and I started talking to Mike, and he's a really brilliant guy, and we started just thinking what we might do. And we set out to make mouse models of autism with the same copy number change, in this case a deletion. And a lot of my colleagues, they knew me from the cancer stuff, and that was really great. And I think a lot of them thought I had lost it a bit. I started working on this other human disease. But for me, it's also a genetics problem, and we had the tools to tackle it. And you might sort of agree with them, there was only a single patient that Mike had found. And then it was great when other people, as we were making the mice, which I should point out, I show these, quickly clipping out a piece of chromosome. They're long experiments, but I think really powerful and worth the effort. But as we were making these mouse models, then other groups said, sure enough, 16p11.2, this set of genes on this short arm of human chromosome 16 is commonly deleted in lots of other patients. And so here are some of the other studies that came out. And so we were getting more and more excited, and it ends up being one of the most exciting regions in autism, important in 1 to 2% of patients, and it's one of the most prevalent lesions. I thought it was interesting because this region was often deleted in patients with autism, And the majority of these deletions were not in the parent per se. It could have been in the sperm or the egg. And so in other words, the parents were not necessarily, or usually not affected. And then these deletions were diagnosed pretty early, 1 to 2 years of age in these children. I thought this region was particularly intriguing because it was found deleted in autism, sure, but also the exact dosage seemed important because it was also seen to be duplicated in other neurodevelopmental disease syndromes, for example, schizophrenia. But these were different. Most of these duplications, when there's an extra copy, tended to be inherited, and they were diagnosed much later in life. And so again, we asked what I think is a really straightforward, obvious question. Are these 16p11.2 chromosome changes really causal in autism-like behaviors, for example? And so again, we could do this because we had all the genes nicely lined up that were lined up in 16p11.2 deletions, 27 genes actually on human chromosome 16 deleted in a number of patients, also clustered together in the mouse genome, just like our cancer gene case. And it happened to be a mouse chromosome 7. So the same 27 genes are here in human and are here in mouse. So we could use our chromosome engineering capabilities to then create these mouse models that we think are useful for studying autism. And the exact nuts and bolts don't really matter, but we can use chromosome engineering. We can focus on a precise region. And we basically have 3 steps. We do gene targeting, basically to stick this, what I show here is a triangle. We modify the endpoint, and then we have a second step where we stick another triangle in at the other endpoint. And then we can either get the third step, we can clip out these triangles again in the embryonic stem cells with this magical enzyme called Cre recombinase, clips these out. And then in this case, if one LOX P site, I should say, is this little 34 nucleotide PCR element, happens to be one is on one chromosome and one is on the other, what we can get in this third step, instead of just clipping this out and making the deletion or deficiency, which I show here, we can make the deletion, but we also have the duplication. So this is more of the details of how we can actually make these. Again, we can make mice where we've deleted the 27 gene cluster, and other mice where we've added an extra copy of this 27 gene segment. And this is just the same way. Once we make these cells that have both a deficiency and a duplication, we do this blastocyst injection procedure, and then we have these chimeric mice, and here, these can transmit either the deletion chromosome, this is marking one of the chromosomes, or the duplication. So too few copies of that 27 gene cluster and an extra copy of that 27 gene cluster. So we can look at the result on these individual mice. This was our first success. We were really happy with this because this showed, based on the coat color here, that these mice that are brown are the ones with the chromosome change. And so this was really great, and we had a project. And again, this was a long, several-year experiment to get to this point. So one of the interesting things is the clinicians will always tell us that the major criteria where autism is diagnosed in the clinic is behavior. And that's the first thing the parents notice about the children about 2 to 3 years of age. And so we thought it important to monitor behavior of these animals. We also thought it was really important, again, we have mice and we'd like to learn something, what it might mean to people, but we didn't want to look at the mice with any preconceived ideas of what we think a mouse should look like if it was a correlate to autism, because the diagnosis is so difficult and variable in some cases that we thought we should just take a good look at the animals and see what they had to tell us. And so to do this, we got our hands on some really nice, sophisticated video tracking equipment. And this is shown here. And so basically what we have this is showing a single mouse let's try to go back. I can show you this movie. So this is the system, and what we have here are maybe that'll work. Okay, actually there's 4 cages that we can monitor of individual mice per cage. And there's an infrared camera that watches the mice in the cage, and it watches what they do day and night for weeks at a time. So we're not interfering with the mice, we're simply watching what they do. And then this will tell us about 50 or so different behaviors that are spit out and can be quantitated, what the mice are doing, what they're eating, when they're sleeping, when they're scratching, when they're walking around, et cetera. And we can watch this in the light, and we can watch this rendition. And most importantly, we can have permanent records of this behavior that we can go back and analyze. So this is just showing you a wild-type mouse that we can and how we can analyze it with this expensive equipment. I won't tell you how much. So then the real question is, what can we learn about our mouse models of autism? So what the postdoc who worked on this, his name is Guy, developed is one thing that he wanted to do is place the mice in cages that were much larger than their normal cages. So they had a ceiling that was much higher. And so they had to adapt their skills to learn how to climb on this enlarged cage and really become acquainted with this new environment during the test as we were video tracking them. And so wild-type mice shown here do this quite well. So what you'll see is they start by going to the lowest part of the cage, they come down cautiously with their butts leading, their hind limbs touching down. Then they go higher and higher on the ceiling of the cage, they go way up there, they're really having a great time. They come down in crazy positions, they come down in a more adventuresome pose, and they really learn quickly and adapt their climbing abilities. But what we found in mice with a deletion is they were much more cautious. They were like these wild-type mice in the beginning. They start by cautiously going up to the lower part of the ceiling, and they come down almost always with their hind limbs leading quite cautiously. And then as we monitored, this was over 10 hours that are shown here, different pieces of this video, they really never adapt their climbing abilities to the new cage. So they continue to climb down up to the lowest part, they come down from the same spot, and they never seem to climb as frequently up, and this one's very cautious, and she's really cute. She's really cute. So they're having problems adapting to this new cage. So you can actually have these traces here that are showing, it might be a little hard to see, the blue shows the wild-type mouse, and you can see maybe if my pointer works better. The dark blue shows they're really much more limited. That's the deficiency mouse, where the purple traces show the wild-type mice use the larger expanse of the cage. We also noticed that some of these mice have repetitive behaviors, and so you can see this in the deficiency. This is the one you see here. And this little girl just kept going up on the ceiling. She climbed up there for 9 minutes at a time and stayed and just really loved the ceiling and just went around and around. And you don't see this in the control mice or the mice with the extra copy or these balanced controls. These are all other controls. And she came down for a minute and went right back up on the ceiling. So she was really fascinated with the ceiling. So they had these repetitive behaviors. And then some of these animals developed these stereotypic ways of getting off the ceiling. So they got up there and they didn't quite know how to come down the regular way. So this is actually real time, and it's this slow stereotypic behavior. And really, it's amazing because she repeated this hundreds and hundreds of times over the recording session. And I think you can't quite see it here, but these are different episodes. At first we thought it was a loop or something. And so these mice have difficulties adapting to the new cage, and they have these repetitive behaviors, and some of them have the stereotypic behavior, and we quantitated this in all the mice, so it was quite fascinating. So some of these behaviors that we saw here in the autism models, these deletion models, they were hyperactive overall. A lot of these mice had difficulty adapting to this new cage environment. And what I didn't show you all the evidence for is they had sleeping deficits, which we could see because we could look over sequential dark and light periods over multiples, and they had these repetitive restricted behaviors. We did also classify these mice with an extra copy of this 27 gene cluster, these duplication mice, and for some of the behaviors, they were affected in an opposite way. So these, in general, were less active, and these slept much more, at least in the dark period. So for some of these behaviors, there was one direction of the behavior when these genes were deleted, and the opposite effect when they were duplicated. And so we recently published this work. And really what we want to be able to do is say, okay, we see these unique behaviors in these mouse models, and can we pinpoint then a particular region of the brain? Are there any brain alterations that might tell us more about the circuits, for example, being affected? And so to do this, we had a really beautiful collaboration with Mark Hankelman at the University of Toronto. And what Mark has essentially is a mouse MRI clinic, if you can believe it. And so we took these individual mice, and I didn't even actually mention, we did behavior on like 50 different mice. And each of the mice that went for the behavioral analysis, we then sent to Mark for the MRI analysis. And what he can do is he can do MRI in these mouse brains, and he can pinpoint volumetric changes in 62 different brain regions. So in other words, he can detect if they're larger or smaller in particular models relative to controls. And what he was able to do is pinpoint these 8 different regions of the brain, colored here in these various colors, that were significantly increased in size in the deficiency animals. And also they tended to be smaller, those same structures tended to be smaller in the mice with the extra copy. So again, like the behavior, there was a reciprocal effect in the mice with the deletion and the duplication. And one of these regions, which is this region shown, this big region shown in blue here, it's kind of the middle region, I'm losing my pointer, was the hypothalamus. And the hypothalamus then we looked at in more detail, and this is the small structure here that was, he can basically take it out of the whole structure. And when we honed in on the hypothalamus, what you could see if you looked at the front of the hypothalamus, the anterior to the posterior, what you can see is a sort of surface model by MRI, the blue, and the volumetric changes are shown here in red. And these are the regions of the hypothalamus that are expanded in the autism models. And this is showing a side view of the same thing. And they can actually section through these virtually and get these sort of slices like you would get with classical pathology. So the hypothalamus is one structure that's definitely altered, increased in size in the autism models. Now, we always get, for each of the projects that I mentioned, there's always a surprise, and I never cease to be amazed by these mice and what they tell us. And for this project, the number one thing that surprised me is that half of the mice, half of the autism models, the deletion mice, died after birth, shortly after birth, within 1 to 2 days. Not all of them, I showed you the ones that lived in the behavioral changes in the MRI, but about half of them died shortly after birth. And we think, and we're working on this, is that these pups, shown here is a deletion, an autism model that is lacking. So normal mouse here, you see suckles from the mom, and there's this little milk patch in the belly. You can see it's like a little white reflection. You don't see that in some of the deletion mice. And so what we think is these mice might have problems suckling from the mother. And the reason this is important is because feeding problems have been associated with the hypothalamus, and we have pinpointed the hypothalamus as being one region, this blue region that's increased in size in those mice, these architectural changes. And so what we think are these changes in the brain might be affecting feeding behavior, as well as these autism-like behaviors of hyperactivity, for example, and repetitive behaviors. So we're looking at this more. So what we found is from these 16p11.2 mouse models of autism is that there are these amazing behavioral phenotypes. These animals are hyperactive, they have a difficulty adapting to a new environment, and they have repetitive restricted behaviors as well as sleeping deficits. And these are all features that are described and used to diagnose children with autism. We also found with MRI, there's increased size of specific brain regions. Interestingly, we think is the hypothalamus is increased in volume. And then Importantly, and perhaps may be relevant to the human condition, is about half of these die after birth. And so for this tells us, provides us with functional evidence that 16p11.2 deletion is causal in these types of phenotypes in the mice. And so, you know, the future and what it might bring for this particular project, I mean, there's a lot of things. Whenever we make one discovery, there's 50 more questions, and it's really exciting. And, you know, we have these really robust behavioral phenotypes, brain anatomy phenotypes, and these feeding deficits. That's great. But I mentioned, you know, and we have these amazing models of increased and decreased dosage of 27 genes. So what is the gene? Is it a single gene? Is it 20 genes? We have no idea right now. It could be getting rid of a group of genes is important, and it might be very different from our cancer study where a single gene ended up being the culprit. So we'd really like to pinpoint the gene or genes responsible. And the way we're doing this is showing the full deletion here, the 27 genes that group up in here. And we know we have these robust phenotypes in behavior and brain architectural changes. Now, if we make mouse models with smaller deletions, for example, will any of these recapitulate? Will they have the same behavioral phenotypes or the MRI? In other words, can we hone in on a particular region? If this particular region, then it would tell us there's about 6 genes involved, and then we can go in and try to study those in detail and have fewer to deal with. So that's one question. What is the gene or genes? Are these actually behavioral phenotypes and brain anatomy and these feeding deficits, are they functionally related? And again, we want to get at the mechanism. What are the nuts and bolts of what's happening here? And of course, can we use the mouse models to improve the mouse behavior, for example? And also, how can we relate this back to the human condition? And I think these are questions for the future that we are very interested in, and also looking at the brain circuitry. So thank you very much, and I'll be happy to take questions.